Okay. Fingers crossed. I did a class this morning where the first 40 minutes had no audio. So this should be fine. If not, take good notes. This looks familiar because we ended with this last class. On Friday, we had introduced the idea of homeostasis and how we maintain various characteristics within our ideal range through use of feedback loops. And so a general feedback loop is shown here. Something is sensed, there's a signal. Something senses that signal, the receptor. The receptor gives information to the controller that compares what's going on to what should go on. And then if those are different, the effector will bring about a change. We'll return something to normal, we'll change something to be able to keep us within that narrow ideal range. Something that we'll see a few times in this course and as we talk about nervous signals coming up are these terms. Input signals in this loop or signals in the nervous system. Input signals are afferent and output signals are efferent. Here that's easy. The effector carries efferent signals. They are signals going to a location to accomplish a goal, to do work. The afferent signals are returning from elsewhere in the body, bringing information back with them. Afferent and efferent are often confused. They're easy to interchange. They're one letter different. There are a couple mnemonics when we get to the nervous section that will help you differentiate what direction each type of information is taking. But for now, we'll leave it at this. Input signals to this processor are afferent, output are efferent. So an example that I mentioned a few times last class was that of blood pressure. I'm going to use blood pressure as an example of a negative feedback loop. So on the last slide, we talked about feedback loops in general. Something is compared and an action happens. A negative feedback loop turns down or diminishes. It responds in opposition to a signal. A negative feedback loop. So the thing that's being measured, the stimulus, is blood pressure. Blood pressure is the subject of our negative feedback loop. And the sensors that monitor blood pressure are cells that are specialized called baroreceptors. Receptors in the name, it's sensing something. Baro is the prefix indicating pressure. Barometric pressure. Baroreceptors monitor pressure at special locations in the body, wherever pressure needs to be maintained within tight ranges. And the way they do this is by firing regularly. They send a signal to the brain, the speed of which is proportional to pressure. So normal pressure has a certain frequency or a certain firing rate. But if blood pressure goes up, these baroreceptors are relaying the information to some processor in the brain that's saying, okay, this is the rate I'm expecting. If it's faster than normal, pressure's too high. It could be dangerous for some delicate tissues. It could be dangerous if you have some kind of artery diseases. So we don't want pressure to be too high. The effector sees that fast firing rate and creates a plan. That plan can involve many different organs. It doesn't have to only be the arteries. It can be the arteries, it can be the heart, it can be different aspects of the heart. It can make the heart beat less quickly. It can make the heart not beat as strongly or as forcefully. It can tell all the arteries to relax and open up. All of those things will help to bring blood pressure back down. So this is a signal that's in opposition to the original stimulus. Overall then, whatever the plan happens to be, whatever organs are involved, the net result should be pressure goes back down. That rate of firing will slow if the plan works. The baroreceptors will then relay that information again. The processor will say, okay, yeah, the plan worked. I don't have to do anything else. We're good for now. Or maybe it didn't work well enough. It only brought it down a little bit and more actions required. The point is, this feedback loop operates continuously until the error is resolved, until the stimulus or the signal is brought within normal ranges. And this is how the large majority of regulation, homeostasis, is maintained within the body, through negative feedback loops. These are by far the most common methods of regulating homeostasis. The opposite is a positive feedback loop. It's the opposite of the example that we just saw. Instead of the plan being created to go against the initial signal, positive feedback loops magnify the initial signal. Low battery. We'll do it live, that's fine. Positive feedback loops magnify the initial signal. Let me just make sure that those of you in the back can still hear me. Yeah? Okay. That's the nice aspect of these auditorium seats. My voice carries up pretty well. Magnifying the signal is not normally something that we want to do. If we're outside of our ideal range, we don't want to go more outside of our ideal range. Yet positive feedback loops achieve just that. And it's in very special situations where it occurs. Pregnancy is one such situation. A positive feedback loop magnifies the signal until the signal disappears. What's the signal? The signal is stretch at the cervix. When the baby is positioned and ready to be born, it's pressed against the cervix and stretch is registered. So contraction seats the fetus at the cervix and stretch is registered. The amount of stretch activates this positive feedback loop proportionally. A little bit of stretch will start this process. More stretch will accelerate this process. Lots of stretch turns this positive feedback loop on maximally. So more stretch occurs, we release what's called oxytocin in the body, which is often referred to as the cuddling hormone. It serves very specific purposes. In this case, oxytocin makes the uterus contract. And you can imagine the uterus contracting around the fetus increases pressure, tries to push the fetus out, creates more stretch at the cervix. Oxytocin reinforces this cycle. Stretch increases, more oxytocin is released, more pressure occurs, more stretch occurs. The only way that this signal ceases is when stretch disappears or is absent. That happens when the baby is born. Until the stretch is released, when the baby passes through the birth canal, there's no more pressure on the cervix, there's no more stretch to be sensed, oxytocin doesn't need to be released anymore, and the positive feedback loop ends. So two complementary ideas. Both of them are loops. A plan is created and carried out based on the information from a receptor. Negative feedback loops work in opposition to the signal to bring it back in line. Positive feedback loops magnify the signal until something catastrophic happens and the stimulus is gone. Now that ended our brief section on homeostasis, and I wanted to get through that on Friday because this is a natural break point. Let me just check in and make sure that that made sense. The idea of homeostasis being some ideal range, we can move in and out of that range for a little bit. The further we move away, the greater the attempts to bring us back within that ideal range. If we go too far, you can die. Temperature is a great example, blood pressure, hormone concentrations, any number of things are regulated by the negative feedback loop and some by the positive feedback loop. I would normally do a brief recap and some questions with Turning Point, although I know that a number of you still have a few issues getting the codes delivered and the codes input. Most of you have it already, which is good. I can see who's subscribed. We will do some questions on Thursday, so try your best to resolve those issues beforehand. I've responded to most of you, but I know some are slipping through the cracks. There are a lot of emails. If I haven't replied, I will shortly. What we want to turn our attention to now, the main focus of today's lecture, is on tissue, types of tissue, and how cells are organized to make that tissue. What are some common characteristics of cell organization within a tissue? Types of tissue. There are some 200 plus specific types, but broadly categorized into 4 main categories, and we'll talk about those today. Tissue is about the basic level, the most basic level from which we'll build up in this course. With few exceptions, we only go down so far as the tissue level. Sometimes we'll talk about individual cells. When we talk about neurons, we'll talk about individual cells in the coming weeks. But for the most part, our concern, our focus is on tissue. So types of tissues. Tissues, you'll recall, are made up of conglomeration of cells. Cells are ordered in layers and grouped together to create different types of tissue. It's what kind of cells, in what configuration, how they're ordered together that determines the type of tissue. Now, on a cellular level, most cells look very similar. Some contain more stuff, some contain less stuff, some contain hard stuff. But when you put them together is where you really start to see the phenotypic or the large-scale differences. Bone tissue is different than muscle. Adipose tissue is different than a nerve or a neuron. Tissue expresses those differences. You'll look at a lot of pictures like this in lab next week. You all got that reminder email? No labs this week, as the people on Monday have figured out already. You'll look at a number of these slides to figure out what kind of cells are involved in this type of tissue. And generally, you'll see what looks to be disoriented, many shapes and forms that aren't really structured properly, but if you look closely, you can see a nice, fine, structured order along the border of this aspect of the tissue, for instance. This is an image of cervical tissue, again, since we're on theme. You can see the nice, normal, what's called epithelium, and we don't know what that is yet, but we'll see eventually. And I highlight this because this is a great example of where order, or where structure dictates function, or where structure and function are tightly related. Normal, ordered epithelium functions properly. This is abnormally structured cancer tissue. You can see the cells have proliferated. Some of them have skewed shapes. And the function of that tissue is also compromised. So you can tell a lot about a tissue by looking at its shape, looking at its structure. So let's consider tissue, a group of cells organized together with a common goal. These usually arise from very similar origins in the embryo. The cells might not be the exact same type of cell. You might have a muscle cell in close proximity with an adipose cell, and that's dictated by the needs of the tissue. All of those cells grow and differentiate together to form a tissue. And the tissue has some specialized activity. We are broadly going to break tissue types up into 4 categories. There are 4 main categories, each with their own subcategories, but for our introductory purposes, there are 4 basic types of tissue. And these tissues mix and match to form all of the organs and structures in the body. Only 4 types. What are those 4 types of tissues? This figure taken from your text shows them nicely. Epithelial tissue is one of the most common that you'll spend a lot of time looking at in lab. And this epithelial tissue, also called epithelium, we saw it on the last slide, is typically the tissue that lines surfaces, that covers structures. The outside of the body, internal organs, cavities within the body are lined with epithelium. They are a covering tissue. They look like this, and you'll see many examples in Lab 1 under the microscope. It sounds boring, lining epithelium, but it has a lot of really diverse functions depending on how it's organized, as you'll come to see. The second type of tissue is connective tissue, and you can kind of think of this as the scaffolding of an organ. The scaffolding of an organ. It provides structure and protection. It connects different areas of an organ together. It helps to anchor them, provide resilience. Connective tissue. It looks a lot less ordered than epithelium, as you can see here on the bottom right. We'll talk about some differences coming up in a few slides. Cartilage is a connective tissue. The compact bone or some of the tissue making up synovial surfaces are connective tissue. A third type that we're not going to look at a lot in lab, unfortunately, is one of my favorite types, muscle. Muscle as a tissue has a very unique role in the body. Muscle is excitable. It can be prompted to do something. It can respond to a signal. Epithelium and connective tissue don't do that. Muscle can. What it does is it shortens. It creates force. It shortens in length. And through that action, we are able to move around. I'm able to speak words to you, and you can hear them. That's largely due to the action of very small muscles in your ear. So muscle tissue has a very organized, striated look, which simply means stripy. We'll see why that is when we get to that section. It's excitable. It can generate force. And lastly, nervous tissue it's the information superhighway of the body. Nervous tissue is also excitable. It can turn on or off. It can relay signals, often to muscle, via what are called action potentials. Action potentials. And that word might not mean a lot right now, and it shouldn't, but action potentials are the signals. They are a burst of electricity, repeated bursts, large bursts. Action potentials are the things that convey information. So last two are excitable, first two are not. These are the four main types. We group them together in various combinations to create different organs in the body. The heart, bones, liver, spleen, pancreas, skin, lungs, anything you want. These four tissue types. Now, I think it's important, before we get too deep into how cells are organized within tissue, to understand how cells connect. How do cells connect? Do they move around? Is it like a bag of oatmeal with everything moving around inside it? No, there are junctions between cells that anchor them to each other and anchor them to specialized scaffolding structures. And cell junctions are really unique because they also will give tissue some of their intrinsic characteristics. We don't often think of cells being connected to anything. When I first got to A&P, I thought, well, cells are all clustered together and the neighboring cells keep the other cells in place. It's the press of all the cells together that keep them in place so they don't move around. But that's not even close to what happens. There are many different types of junctions or points of connection between cells, and it's worth paying attention to 5 main types. I'm going to emphasize 2 of them because we'll talk about them repeatedly throughout the course. The first, you can see at the top here, this braided network of proteins creates a tight junction. And a tight junction, like its name implies, brings the cells together in very close proximity so that it seals the space between the two. It seals the space between the two tightly. These proteins woven together are like the upper seal on a Ziploc bag. Hopefully it's intact so that when you turn it upside down, your spaghetti sauce doesn't fall out. It's a tight junction. And sometimes we want the area between cells to be sealed. We want to keep some things on one side and other things on the other. It's very important in the brain. We'll see that when we get there. So that's one type of junction, a tight junction. Adherens junctions are somewhat similar, but less watertight or less airtight, put it that way. These tend to be a band that circle around the top of the cells and give them shape, that help them maintain their round, open shape. They connect to other adherens junctions on other cells, on neighboring cells, and they help to give it structure. They encircle and anchor the tops of the cells. But things can pass through. It's still hard, but some things can pass through. In the side, down here on the right-hand side, between these two cells on the right-hand side are these little circular, hairy-looking structures that I think of as spot welds. These are desmosomes that anchor at one point two neighboring cells. It's not a band, it's not a strip. Things can move around this anchor point, but it helps to keep these two neighboring cells oriented. They're side by side, and they stay that way because of these desmosomes. You'll also see mention here, and I'm not going to go into detail on this, but within each cell there is a skeleton. Just like within your body, the bones create the skeleton. In each cell we have what's called a cytoplasm. skeleton. There are proteins that run all the way across the cell, up and down, left and right, and this is how things get transported within the cell. We've got a fantastic video at the end of second semester, if you can wait that long, that shows the cytoskeleton in breathtaking detail. These spot welds also serve as connection points for that skeleton. So you might have one long strand running from one side to the other that allows fat or carbohydrates or different hormones or proteins to be moved within the cell. That's what these hairy looking structures on the outsides are for. They connect to the cytoskeleton. Number 4, or really 3B, is very similar to the last type. It's also a desmosome, a spot weld, but this is a special type of spot weld in that hemidesmosomes anchor the cell to a basement membrane. Not to a neighboring cell, but to some other flat surface, a basement membrane. I'll come back and explain what that is shortly. Hemi simply means half a desmosome. So you see this half a desmosome like we saw on the right side of the cell. Looks very similar, but the point underneath, the inferior half, is different because it's connecting to a membrane. It's connecting to a hard, flat surface, not to another cell. Hemidesmosomes specifically anchor to the basement membrane. And lastly, the junction that's not a junction, gap junctions. Gap junctions are very cool. This is how all of the nervous signals in your body get sent. The transmission of signals through gap junctions. And gap junctions, again like their name would imply, are porous. Two neighboring cells are connected together with these small pores that allow things to filter through. Fluid, ions, some small molecules. The contents of the two cells bridge through gap junctions. Isn't that bad? Don't we normally want cells to be intact and separate? Normally, yes. In the case of nerves and the transmission of action potentials, no. We desperately need gap junctions in place for those signals to be transmitted down the line, distally, throughout the nerves to get to their destination. So we will return to gap junctions a fair bit when we talk about neurophysiology. We'll return to tight junctions when we talk about neuroanatomy in the brain. These other ones we don't really return to. 1 and 5, I would say, are the most important types of junctions on this slide. So cells aren't only put in a bundle, left in a heap, and kept in place by the other cells around them. They are purposefully anchored and arranged in a way that suits the tissue. And how they are arranged gives that tissue some characteristics. If we're using a lot of tight junctions, that tissue is probably a barrier. If we're using a lot of gap junctions, all the cells in that tissue can communicate easily, all due to the type of junction being used. Now let's dial it back from our in-depth look at junctions and then talk about the tissue level again. Here, I want to compare briefly connective with epithelial tissue. These are the two prominent types of cells or types of tissues that you'll look at in lab. They are also often present together in the body. Epithelial tissue is the covering tissue. Connective tissue is the scaffolding. The way we combine these two together dictates the type of tissue we create and the type of organ that results. Epithelium often sits on top of connective tissue. It's not to say they can't occur independently, but they often occur together. And here we have a side-by-side comparison of the two types of tissues. If you were paying attention to the earlier histology slice, you would have noticed both of these together on the slide. I'm separating them out here. You can see a couple differences. On the left is the epithelium, the lining tissue that covers an organ or covers a surface, and on the right is connective tissue. And right off the bat, you can see there's a lot more stuff around the cells of connective tissue. The arrangement's wildly different. The cells within each tissue are the dark points. The dark points are specifically the nuclei. You can see many different nuclei, many different cells sandwiched together on the left. And on the right, they're dispersed. There's very few, and the area between them is full of a lot more stuff. Maybe it's fluid, maybe it's proteins, maybe it's some adipose, but there's stuff between the cells. There's some blood supply, there might be some nerves, there might be structural proteins. It's wide open. There's more stuff in the space between cells. And that space has a special name. It's outside of the cell, so it's extracellular. And that stuff makes up the scaffolding, so we call it a matrix. The space around the cells is the extracellular matrix. So the orientation, the distribution of cells, wildly different. We have a lot more space in connective tissue for blood supply. Epithelial tissue has very little blood supply. There's not enough room. The cells are sandwiched together so tightly, we don't have a lot of projections of blood vessels into the tissue. Epithelium is said to be avascular. It's not vascularized. Connective tissue, highly vascular. Don't tissues or cells need blood supply to survive? Absolutely. Epithelium is often in close proximity to connective tissue because it carries a blood supply with it. Things can still move and diffuse around, but there's no direct blood supply to the epithelium. And what's not shown here as a difference is epithelium's always, or usually, on top of connective tissue. These two occur together. They are often in close proximity. They're intimately related, one on top of the other, epithelium on top. Perhaps a better word is more superficial or closer to the surface, because we've learned top doesn't mean very much in anatomy, right? So these are complementary structures. Let's look at how they're arranged. And I'll come at it from the point of view of epithelial tissue first. So from the surface, what are some general features of this type of tissue? The lining tissue, the covering tissue. How is it associated with connective tissue? And you can see a good example of it in the slide behind on the background. We'll come back to this after I go through these details, but you can see epithelium, various degrees of epithelium, and some connective tissue here and, no, just here. So epithelium is arranged with cells that are densely packed together. We saw that on the left-hand side of the last slide. They're usually arranged in sheets. They could be thick, they could be thin. There could be many sheets, but they are densely packed together. As such, many junctions are present. Many cells, many points of contact, many anchoring points, many different junctions. And it doesn't only have to be one type of junction. All of the types of junctions can be present, or some. Many types of junctions. They will eventually attach to a basement membrane. That flat surface that anchors the cells, where the hemidesmosomes anchor the cells, it's called a basement membrane. Just like the basement is the lowest point in a house, the basement membrane is the lowest point of connection for the epithelial cells. Because they're so densely packed together, because there's so little space, we've already indicated there is no blood supply, but there is a nearby blood supply, which happens to be good enough. The things that epithelial cells need can get there by diffusion. Being a covering tissue, being a lining, this is often the point between the inside and the outside of the body. The inside of a tissue and the space or cavity within the body. That is, it's a point where it will come into contact with other things. The skin, for instance, comes into contact with anything you hit, touch, wear on a daily basis. Those serous membranes that we talked about last week that line the inside of the cavities come into contact with the organs that are held in those cavities. The lining of the intestines comes into contact with food. So this tissue is always being roughened up. It's always in contact with new other things. It's cut, there's friction, there's abrasion. It's always being rubbed and damaged. As a lining tissue, it's always being damaged. And so mitosis occurs frequently to replace the damaged cells. Skin is always regenerating. Linings in the intestine are always regenerating. Dead cells disappear, they slough off. Dust in your house is just dead skin cells as they are replaced by frequent mitosis. So these are general features of epithelial tissue, and you can see a schematic of what it might look like shown here. These pink columns are epithelial cells making the epithelial tissue. This is lining the basement membrane, this double-layered blue surface, and below it we have connective tissue as the scaffolding. You'll often see tissue depicted in such a way, certainly epithelial tissue. From the top down, the outside surface pointing away from the body is called the apical surface. This surface can point out to the room if it's on the skin. It can point into the stomach if it's the lining of your stomach. It can point into the abdominal cavity. It could point into a blood vessel. It's pointing away from the body into a space. That's the apical membrane or the apical surface. On the other side, in close relation or proximity to the basement membrane is the basement or the basal surface. We'll often use those two terms interchangeably. The basal surface, the basement membrane are very closely related. The bottom pointing towards the body. And that basement membrane itself, you can think of as it's a structural sheet. There are various sticky proteins and structural proteins that make this flat surface where the epithelial cells can anchor, provides an attachment point to connective tissue, and it always intervenes. It's always found between the layer of connective and epithelial tissue in an organ. Now, I won't go into detail about how the basement membrane is formed. It's not important right now. Know that it's there. Know that it anchors the epithelial cells to the connective tissue. And before we leave, notice the blood supply. It's not into the cells of the epithelium. The blood supply ends right here. This is a blood vessel. You can see a red blood cell in the blood vessel. It probably U-turns and heads back down. Any nutrients required by the epithelium will diffuse from this blood vessel through the connective tissue, through the basement membrane, into the cells. That's how these cells get the nutrients they need. It's not delivered directly, it's delivered by diffusion. They do, however, have nervous supply. This yellow squiggly. Nerves are always colored yellow. Arteries are always colored red. Veins are always colored blue or purple. Yellow indicates there is nervous innervation to the epithelium proper, which is good for a variety of functions that we'll see coming up. Let me just finish one more slide, and then we have one announcement before we call it for the day. Yeah, question in the back. It is pointed towards an opening. Yes. It could be the surface of the skin. It could be into the abdominal cavity or into a blood vessel. Absolutely, depending on the type of epithelium. And that might be slightly confusing at first because we're talking about blood vessels supplying the tissue making a blood vessel. That absolutely happens. The musculature around a blood vessel needs its own blood supply. So this does happen. But the top goes to a surface or an opening. Good clarification. Bottom goes towards more tissue into the body. Does that make sense? Let me introduce this concept quickly and we'll pick it up in class on Thursday. So far, we've seen very simple organization. The epithelial cells are lined up. They're columns, they're single cells, but the type of tissue varies depending on the shape of those cells and their arrangement. There are a number of different arrangements. These are indicated here. Simple is one layer. Pseudo-stratified makes it look like there's multiple layers, but they're really just garbled cells. Stratified are multiple, ordered, distinct layers. Epithelial tissue can come in any of those forms. And it can have any number of these different shapes. Squamous cells are generally very thin. Still an epithelial cell, but very thin. Cuboidal, like the name implies, are shaped like a cube, and columnar, like a column. Depending on what combinations of cell shape and cell arrangement you have, dictates the function of that epithelium. We'll talk about how these are arranged a bit more in class on Thursday. We will talk about, or you'll see it in lab, but right now there is one quick announcement from the HKIN Society, briefly before we call it for the day. I assume that's you ladies at the back. If you want to do your little spiel, I have the slide that you sent me shown here. This is Regan and Brenna. Is that correct? Okay. Regan and Brenna, this is the first year HKIN class.
Thank you so much for letting us come in.
You're very welcome.